Just like the primary structure of a protein is determined by the amino acid sequence, the primary structure of a nucleic acid is determined by the sequence of nucleotides. And nucleotides are going to react through a series of condensation reactions that follow a very specific pattern. So there is a directionality to a nucleotide sequence, and the first nucleotide is always going to have what's considered a free 5' carbon on the pentose. And its 3' hydroxyl is going to react with the phosphate group of the next nucleotide in the sequence. So there has to be a little bit of acid present, and that acid is going to become part of the water molecule that is released during the condensation reaction. And what we see is that when two nucleotides link together, the first nucleotide in the sequence is going to have a free phosphate on the 5' carbon of the sugar, and its 3' hydroxyl group will react with the phosphate group on the next sugar that is, of course, connected to its C5. So there's going to be this 3' to 5' linkage between all of the sugars in the nucleic acid, and these sugars are going to be connected by this phosphate group, and this linkage is known as a phosphodiester. Once the nucleotide sequence ends, or the nucleic acid ends, then the last nucleic acid is going to have a free 3' carbon, is what it's known as, and so that hydroxyl group remains unreacted to a phosphate. So if we look at the backbone of a nucleic acid, we see this common structure of phosphate, sugar, and of course these phosphates are connected between the 3' and the 5'. And this gets sort of uninteresting and actually unnecessary. What we really care about is the identity of the bases that are bonded to the ribose or the deoxyribose. So when we are signifying a nucleic acid sequence, we just merely have to represent the bases. And oftentimes we will signify which base is first in the sequence by calling it the 5' because that 5' carbon is free or has the phosphate that's not reacted, and then end it with the 3' because that 3' carbon of that last sugar is also not reacted. That hydroxyl group still remains on that carbon 3. So let's look at sample problem 11.4, nucleic acid formation. Draw the RNA dinucleotide formed if two adenosine monophosphates undergo a condensation reaction. Label the 5' and 3' ends of your structure and identify the phosphodiester bond. So remember, the first adenine is going to react with its 3' and it will have a free 5' end, meaning this phosphate group on the carbon 5' is not reacted. Its hydroxyl that used to be on this 3' reacts with the phosphate of the next adenosine monophosphate. And here is the phosphodiester bond that connects the 3' and the 5' carbon. And then the second nucleotide has the free 3' as this hydroxyl group has not reacted. Let's look at sample problem 11.5 that asks us to write out the nucleotide sequence of this structure that is illustrated. So we're going to go from up to down. We notice that the top base, or the top nucleotide, has this free 5' carbon, so this is the first one, and the one at the bottom has the free 3' carbon, so this is going to be the last one. So we just have to identify the bases. So if we look at the first base, that is going to be guanine, or G, and if we look at the second base, that is going to be cytosine, or capital C. If we look at this third base, that is going to be uracil, or capital U, and then the last base is also guanine. So you could just write GCUG, since it's always assumed that from left to right we start with a 5' end, or you could specify 5' GCUG 3'.